Today we are going to start a new chapter, Molecular Basis of Inheritance. In this particular chapter, we will cover every topic prescribed by CBSE, and that will be helpful for any class 12 student of India. Before starting, we have to say that the chapter is very long, and that's why we have divided the chapter into some small part. You can check it from the playlist to complete the entire chapter. Let's begin. The name of the chapter is the molecular basis of inheritance. What is inheritance? We need to know the term. We need to know the term first. Inheritance is the process in which characters are passed from one generation to another, from parent to their offspring, their child. This process is called inheritance. But how? To know this, you need to recap the previous chapter, Principle of Inheritance. But in this particular chapter, we will only discuss about the molecule that is responsible for inheritance. As we know what is the genetic material, what is the molecule responsible for inheritance, but it was not that easy. Scientists have been researching for many years to identify the genetic material. In 1869, scientist Friedrich Mischer isolated a substance from white blood cell, and he found that this substance contained carbon, hydrogen, nitrogen, oxygen, phosphorus. He thought that the substance is protein, but this substance contained phosphorus, and this should not be present in protein. And later he found this particular substance into the nucleus of every sample that he studied, and that's why he named it nuclein, as it is present on nucleus, and that's why he is credited for discovering nucleic acid. Then in 1928, Griffith, a British medical officer, was working with Streptococcus pneumoniae, that the bacteria that is responsible for pneumonia disease. He was working with two strains of Streptococcus pneumoniae. One is S and another is R. A stands for smooth. That means these type of bacteria forms smooth colony, smooth and shiny colony, and another R forms rough colony. This A strain is smooth because it has polysaccharide coat to their outer membrane, but this R don't have this. And another important characteristic is these A's are virulent. But these are virulent, that means it can cause pneumonia. It is harmful, but this R is harmless. That means the bacteria that have polysaccharide coat is virulent, that can produce pneumonia, but those that don't have the polysaccharide coat cannot produce pneumonia. Griffith inject this bacteria into mice. When he injected the S into the mice, he found all the mice were dead. When he inject the R type of bacteria strain into the mice, he found that all the mice were alive. And then he killed the S bacteria with heat. Killed the S type of bacteria with heat. That's why it is called heat-killed bacteria. Heat-killed S. And he injected the heat-killed S into the mice. And he found that the mice were alive. Next, he mixed the heat-killed bacteria, heat-killed S with R, and the total mixture was injected into mice. And he found that the mice were dead. See, the heat-killed bacteria S, the heat-killed S and R are not virulent individually, but when they mixed up, they are virulent. That means something from this heat-killed bacteria are transferred to R and make it virulent. That means S. That something is transferred from this heat-killed S to R and make it S by producing polysaccharide coat and make it virulent, and the mice was dead. Griffith named this transforming principle because it transformed R into S. This transforming principle is very important. In 1930, another group of scientists, Avery and his colleagues, tried to identify the transforming principle. They actually take some A strain and lysed it and collect the cellular component that is called cell extract. They mixed this cell extract to the R type of bacteria that are non-virulent, and they found this R are changed to S. That means this cell extract has something that is transforming principle, and these make the R into S. It is confirmed that this cell extract has transforming principle. Avery and his colleagues knew that the cell extract has some macromolecules that are actually the transforming principle. What were the macromolecules? Those are protein, polysaccharide, DNA, RNA. They knew that one of them are transforming principle. That means the genetic material. They use enzymes that only degrade the particular macromolecule. Suppose they use protease into cell extract. all the protein of this cell extract will be degraded, and if this is mixed with R, and if the transforming principle was protein, it cannot change the R into S as it is degraded by protease. Simple. But they found only it is possible when DNA is degraded. That means when they use protease into cell extract, the result is same. R is transformed to S. When they destroy a polysaccharide, then also the R is transformed to S. But when they use DNase, these R is not transformed into S. So they concluded that DNA is the transforming principle. But that time, most of the scientists thought that transforming principle was protein. In 1953, Hershey and Chase published a paper that provided more evidence that the DNA is the genetic material. We will discuss about their experiment in the next class. Now tell me the name of the bacteria that was used by Griffith. Comment your answer.